So, I am Rabin Debnath from Institute of Pharmacy, Assam Don Bosco University. I am here to share my presentation on the B Pharm second semester Unit 1 topic, which is biomolecules. So, today we are going to cover here the introduction of biomolecules, classification, chemical nature, and biological role of carbohydrates, and also we'll study about lipids. So, biomolecules. Biomolecules are the molecules that occur naturally in living organisms. Biomolecules include the macromolecules like proteins are there, carbohydrate, lipids, nucleic acids. It also includes small molecules, means along with the macromolecules, which are proteins, which is made up from amino acids. It also includes small molecules like primary and secondary metabolites and natural products. Biomolecules consist mainly of carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. These are the elements which are seen generally in biomolecules. Biomolecules are very large molecules of many atoms which are covalently bounded together. Moving to the next slide. Biomolecules are further classified into two types, which is micromolecules and macromolecules. Micromolecules are small sized, low molecular weight between 18 and 800 Dalton found in the acid soluble pool. Examples are minerals, gases, water, sugar, amino acids, which is the building block of proteins, nucleotides, which is the building blocks of nucleic acid. On the other hand, macromolecules are the larger molecules. These are made from the micromolecules, which molecular weight is above 1000 Dalton, found in the acid insoluble pool. Examples here are carbohydrates, lipids, proteins, and nucleic acid. So, biomolecules, as I've said, it is either micromolecules or macromolecules. Micromolecules is the smaller one, and macromolecules are bigger. Micromolecules build, come together, join to make the macromolecules. Now, moving to the third slide, major complex biomolecules of cell. So here are the major biomolecules. For example, polysaccharides. Polysaccharides, the building blocks are monosaccharides. Many monosaccharides come together to form the polysaccharides. They are joined together by glycosidic linkage. And the major function is stored form of energy. Proteins are made up of amino acids. It gives the basic structure of cell and also the basic function of cell. Lipids are made up of fatty acid and glycerol. This is the storage form of energy to meet long term demands. DNA, also known as deoxyribonucleic acid, the building blocks are deoxyribonucleotides. Major function is hereditary information. RNA, which is ribonucleotide uh, uh, ribonucleic acid. It is made up of ribonucleotides. Major function is protein synthesis. Next, carbohydrates. Starting with the carbohydrates are the most abundant naturally occurring carbon compounds in the nature. It is made up of, mainly it contains carbon, hydrogen, and oxygen, which is present in the ratio of 1 is to 2 is to 1. And the general formula is CnH2O whole N. Since most of the carbohydrates are sweet in taste, hence they are also known as sugar. Examples are sucrose and lactose. Carbohydrates are the very good source of energy because they helps in producing ATP. Carbohydrates or polysaccharides are long chains of sugar. They are made from monosaccharides. Monosaccharides are simple sugars that compose of 3 to 7 carbon atoms. Next, they have a free aldehyde or ketone group which acts as a reducing. In carbohydrate, there is a free aldehyde or ketone group which acts as a reducing agent. Hence, they are also known as reducing sugar. Disaccharides are made up of two monosaccharides. It is a part of oligosaccharides. The bond shared between the monosaccharides are known as glycosidic bond. Monosaccharides and disaccharides are sweet, crystalline, and water-soluble substances. Polysaccharides are polymers of monosaccharides, means they are made up of many monosaccharides. They are not sweet and complex carbohydrate, they are insoluble in water and are non-crystalline in nature. Examples are glucose, fructose, which are monosaccharides, sucrose, maltose, which are disaccharides, and starch and cellulose, which are polysaccharides. Functions. General formula, as I've already told, which is CnH2O whole N. Chemically, carbohydrates are defined as optically active, means which can turn the plane polarized light, rotate on plane polarized light. Is are optically active polyhydroxyaldehydes or ketone, or the compounds which gives such units on hydrolysis. Carbohydrates are chief energy source in many animals. They are the main energy source. They are instant source of energy. Glucose is broken down by glycolysis process into your pyruvate, and which is further converted into acetyl coenzyme A and enters the Krebs cycle in order to yield ATP. Glucose is the source of storage of energy. It is stored as glycogen in animals and starch in plants. Stored carbohydrate acts as energy source instead of protein. Carbohydrates aids in regulation of nerve tissues and are the energy source for brain. Moving forward, carbohydrates classification. Here we have monosaccharides. Monosaccharides are the basic unit which cannot be hydrolyzed into smaller units. They are further classified based either on the number of carbons or type of the functional group present. Shown below are glucose, galactose, and fructose. See, glucose and galactose are epimers of each other. They have the same formula, same almost same structure, but they are epimers. And fructose have the same formula. All of them have a formula C6H12O6, but different structure. Whereas in case of glucose and galactose, they are epimers of each other, means only at one position there is a difference. Then oligosaccharides. Under oligosaccharides, it consists of disaccharides, trisaccharides, and tetrasaccharides. It can be further hydrolyzed into simple monosaccharides. Polysaccharides, as we already discussed, are non-crystalline, non-soluble in water. It is tasteless. On hydrolysis, it gives many monosaccharides. Example is starch, cellulose. Now monosaccharides. Monosaccharides are the simplest group of carbohydrates. In Greek word mono is one carbohydrate and are often referred to as simple sugar. They have the general formula of CnH2O whole N, and they cannot be further hydrolyzed because it's the simplest form. The monosaccharides are divided into different categories based on the functional group and the number of carbon atoms. If we talk about the functional group, first is aldoses. When the functional group in the monosaccharides is an aldehyde, they are known as aldoses. Example, glyceraldehyde and glucose. Whereas ketose means the functional group is a ketone group. They are referred to as ketosis. Example are dihydroxyacetone and fructose. Monosaccharides, based on the number of carbon atoms, it can be further classified, which are triosis means which have 3 carbons, tetrosis means 4 carbons, pentosis means 5 carbons, hexosis means 6 carbons, heptosis means 7 carbons. Among this, pentosis, which is the ribose, here which can be seen in the ribose sugar, which is present in the DNA. Now, this we can see as representation on based classification of monosaccharide based on the number of carbon atoms and based on the functional groups. Now, glyceraldehyde, the reference carbohydrate. Glyceraldehyde or the triosis is the simplest monosaccharide with one asymmetric carbon atom. It exists in two stereoisomers and have been chosen as the reference carbohydrate to represent the structure of all other carbohydrates. Means this is the reference which shows the structure of all other carbohydrates taken as the reference. Here we have D form and L form. These are the two isomers, are mirror images of each other. The spatial orientation of H and OH groups on the carbon atom C5 for the glucose, that is adjacent to the terminal primary alcohol carbon determine whether the sugar is D or L isomer. Means here D and L isomers. For example, if you say about dextrose, D form of dextrose is also present. If the hydroxy group is on the right hand side, the sugar is of D series, and if the hydroxy group is on the left hand side, then it belongs to the L series. Okay. as we can see here, D-glyceraldehyde, as the hydroxy group, the second one is on the, if you see this one is on the right hand side, is called as D-glyceraldehyde. If it is on the left hand side, which can see in the right hand side image, it is L-glyceraldehyde. Similar to glucose, here the fifth carbon. If you start from here, this is the functional group 1, 2, 3, 4, 5. Fifth carbon hydroxy group is in the right hand side, so it is D-glucose. If it is in the left hand side, it is L-glucose. Next, epimers. As I already discussed, glucose and galactose are epimers of each other. If you see the structure of galactose and glucose, we can see that in galactose, the hydroxy group is on the left hand side, and in case of your D-glucose, it is on the right hand side. Other structure is similar and they have the same formula. Only structurally at one position they are different. Now if you see formation of hemiacetals and hemiketals. Actually, aldehydes and alcohols, when they react together, they form a hemiacetal. The hydroxy group of the monosaccharide can react with its own aldehyde or ketone functional group to form a hemiacetal. If it is a ketone, then it is a hemiketal. Thus, the aldehyde group of the glucose at C1 reacts with alcoholic group of C5 to form two types of cyclic hemiacetals, namely they are alpha and beta. Now, derivatives of monosaccharides. If you see the derivatives of monosaccharides, here is deoxy sugar, deoxygenation of the ribose product, deoxyribose present in DNA, which is the structural component of DNA. Next, amino sugar. When one or more hydroxy group in the monosaccharide is replaced with the NH2 group, it forms an amino sugar. Example, glucosamine. Thirdly, we have sugar acids. Oxidation of the CHO, which is the aldehyde or the hydroxy group from sugar acids. Ascorbic acid is a sugar acids. So, oxidation of the CHO or the hydroxy group forms your sugar acids. On oxidation become acids. It is also, for example, ascorbic acid is a sugar acid. Sugar alcohols, reduction of the aldoses or the ketosis, glycerol and mannitol are the examples. Now, oligosaccharides. Oligosaccharides, as we have already discussed, it contains disaccharides, trisaccharides, and tetrasaccharide. It can consist up to 2 to 9 monosaccharides. More than 10 is your polysaccharides. The smallest, for example, here we can see galactose and glucose. Okay, so this is the consist of galactose and glucose. This is a structure of lactose. These two monosaccharides are joined together by your uh, glycosidic linkage. And if you see here, this the numbering starts from this carbon. This is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and this one is the sixth carbon because it consists of six carbon. And if you see here, this carbon, the C1 carbon, is known as the anomeric carbon. And here, if the hydroxy group is on the opposite side as compared to the sixth carbon, then this is known as your trans configuration, and this is known as your alpha. As it is a glucose, so it's known as alpha glucose. If the hydroxy group will be on the upper side and this hydroxy group is already in the upper side, so this is the cis configuration which forms the beta glucose. So this can also be uh, categorized as alpha and beta, the glucose, fructose and galactose, based on the spatial arrangement or the positioning of the hydroxy group of the first position carbon and the sixth position carbon. In case of fructose, it is the second position and the sixth position carbon. So this is the structure of sucrose. Sucrose is made from your fructose and your glucose, and lactose is made from glucose and galactose, and maltose is made from two alpha glucose which are in the this. Here you can see that the direction, they are joined together by glycosidic linkage, and the direction it is represented here, which is 1 to 2. Okay, so alpha glucanopyranosyl 1 to 2 is the direction, beta D fructofuranose. This is the nomenclature. Next, you will see disaccharides. Disaccharides consist of maltose, lactose, and sucrose. It also called as malt sugar, made up of two glucose molecule. As we have already discussed, lactose is also known as milk sugar. It is made up of glucose and galactose. Sucrose is cane sugar that we normally eat in home. It is made up of glucose and fructose. Next, polysaccharides consist of more than 10 monosaccharides. They are also called as glycans, made up of repeating units of monosaccharide which are held together by glycosidic bond. It consists of the examples of polysaccharides are starch, cellulose, and glycogen. So here we can see homoglycans are there, heteroglycans are there, as polysaccharides are also known as glycans. Homoglycans is made up of only one type of monosaccharides. Example in starch, glycogen, and cellulose. Whereas in heteroglycans, made up of two or more type of monosaccharides. Suppose it can be glucose and fructose, like that. For example, in case of starch, glycogen, and cellulose, it is alpha glucose. So with this, the carbohydrate part is covered. The examples of heteroglycans are hyaluronic acid, agar, chitin, etc. and peptidoglycans.